I appreciate I'm the last speaker of today, so I'm going to try and make this as painless as possible. If you guys have any follow-up questions, I'm glad to discuss any details later on. So I work at LabGenius, as Brun already said, and at LabGenius we are combining together synthetic biology, machine learning, and robotics in order to build a platform to design proteins. And I know proteins have been discussed in this room before, but normally when I say proteins, different people have very different concepts in their head. Some of them are very vague. My grandmother, for instance, is really upset with the fact that I'm vegetarian, and she keeps telling me that I need more protein in my life. Well, this is not actually true. I don't, because proteins are everywhere. They're behind all the life on this planet, even in plants. And human bodies, for instance, have about 20,000 different proteins. Because of those, we're here and functioning. But what are those proteins, right? Let's add some context here. So proteins are tiny little molecules of nanometers in size. This big guy here, for instance, is actually a complex of different proteins, and it is 1,000 times smaller than a diameter of a hair. But despite the fact that they're so tiny, they can have various different structures and various different functions. Some of them can move things around, recognize pathogens, emit all sorts of light, just to name a few functions. So imagine we could design these proteins to our own needs. Imagine we can build a molecular machine that would do just what we want it to do. Imagined? This would massively speed up the scientific discovery progress, but also cure a lot of diseases. And every single one of these tiny little molecules could become a billion-dollar drug. How do you design a new protein, though? They're so tiny and they have so many different complex small structures. How do you make a structure that does exactly what you want it to do? Well, the good news here is that if you unfold every single one of those tiny little molecules, they will turn out to be linear chains composed of 20 different types of building blocks or amino acids. So you can take any single protein, be it a protein from your smoothie or a protein from the lab, and write it down in the form of a string that is composed of 20 different types of letters, where every single letter is encoding a specific chemical structure. So you give this string to a scientist, and they will know exactly how to do this string, how to make this string to become a physical molecule. So actually, when you're trying to design a new protein, what you want to find is the string that encodes the structure that does exactly what you want this molecule to do. So the complicated problem of protein design becomes a combinatorial problem. Obviously, the problem here is that the search space is absolutely massive. If you have a protein of 2 amino acids, which is never the case, but just let's stick to it, then you would have 20 to the power of 2 different variants. If you had a protein of 100 amino acids, which is normally shorter than the standard protein, you would have 20 to the power of 100 different variants. And if you had a protein of 27,000 different blocks, which is the largest protein that is known today, then you would get 20 to the power of 27,000 different variants. And just to give you some perspective, the number of stars in the observable universe is estimated to be about, I believe, 10 to the power of 21,000. This is an insane, incomprehensible number. But have all of these proteins been seen in nature? Well, actually, if you think about all of the proteins that have been sequenced, which means that scientists have seen those proteins in nature, they will only constitute a tiny fraction of all of the proteins that exist today. And those guys will constitute only a tiny fraction of all the proteins ever. So they have been proteins before our time, but they haven't survived. And those guys only constitute a tiny fraction of all potential proteins, which is basically an infinite number. So nature hasn't even explored the full potential of protein designs because it's basically infinite. So scientists came up with a very, I think, elegant concept when they're trying to think, to basically comprehend this huge space, which is called protein fitness landscape. And I'm going to walk you through it. It's quite simple, actually, and not really mathematical correct, but it's beautiful. So if you take all of the variants that exist of a protein A and map them out on a two-dimensional surface, where each, could you hear me okay? I don't know. Where each point corresponds to a protein variant, and all of the adjacent points correspond to similar variants. And then the further you go away from the original protein, the more different your protein becomes. Sorry. And then you add another axis that would tell you how good each variant is, right? So you would get a sort of a landscape. This concept is called protein fitness landscape. And what, throughout the history of life, what evolution was doing, was actually trying to navigate through that fitness landscape to the peaks. But the problem with evolution, and Jennifer has talked about it a bit before, before me is that it's quite random. The process is quite random and blind. So the way new proteins are designed is basically introducing random changes and then seeing what happens. If the change is good, the protein will stick around. If the change is bad, it will die out. So navigating the protein fitness landscape using this directed evolution kind of technique is like coming to a city, not having a map, there are no people in the city, and you're trying to find several buildings. How long will it take you to find those? Very, very long. Depends on the space of the city, right? We know it's the infinite space, so forever. What would be a better way of navigating? Well, having Google Maps, right? If we had a map, then we would know exactly where the peaks are on this landscape. We could go to those peaks and we could sample the sequences from those peaks. So we think that the way to do protein design and drug discovery in its whole is to build a map of the protein fitness landscape or protein fitness landscapes, and then basically go where we need to be and sample from those peaks. And the way to do this, we believe, is called a framework. It's called empirical computation. So empirical computation is a framework that is combining both experiments and computers. So you would be basically testing lots of different variants in the lab, and then you would be using the data that you have produced from those tests in order to inform the next generation library design. So empirical computation consists of 4 steps. As I said before, you build the molecules physically, you have to build them physically first. Then you would test those molecules and rank them according to their performances. Then you would use the data that you have obtained from those tests in order to build machine learning models describing the sequence to function relationship. And once you have those models, you can design the next generation in a more informed manner. Not random like evolution does, but more in a sophisticated way. And then once you design the next generation, you can build it and so on and so forth until you have discovered the molecule that you were interested in. And we're, of course, not the only ones who believe in the power of empirical computation, and we're not the ones who discovered this framework. There are other companies in the space as well, and academics as well. But the caveat with empirical computation platforms is that they have to be extremely focused. This is due to the experimental biology design. So if you need to implement experiments, you have to make sure that they're robust and reliable, and it takes you a lot of time and money to do that. And as I said before, we're focusing on proteins. So what is the main challenge when discovering a new protein? Well, normally you will never be asked to design a protein that does only one thing, especially when it comes to drug design. So if you were to discover a new drug and it does exactly what you want it to, but say it has high immunogenicity, you're never going to give it to a patient, right? So imagine you've spent a lot of time discovering this molecule, and 9 out of 10 features are fine, but one toxicity is skyrocketing. Are you going to see this molecule to the clinic? Of course not. This is going to fail. And then a lot of efforts that you have spent developing this molecule can be basically binned. So when I was talking about a protein fitness landscape before, I was actually simplifying the concept of fitness, right? Because actually it's not just one axis, it's a combination of several. It's a combination of lots of different landscapes that are convoluted to compose this fitness. So in order to describe this fitness, what you want to do, you want to deconvolute those unitary fitness landscapes and describe those. So the way it works in our platform, as I said before, we start with lots of different protein designs, physical molecules, and then we develop tests in the lab that would decouple the different properties of the proteins that we're interested in. And then we would use the results of those tests to build machine learning models around those tests, so to link sequence to the properties of interest. And then the final step is to use multi-objective optimization to co-optimize across these different properties together. And then you can inform the next generation protein design or build the molecules. So just to add a bit of resolution to my words, I'm going to talk you through 8 easy steps to implement empirical computation platform for protein engineering. It goes like this. So as I said before, first things first, you have to build a library of proteins physically. So you have to have the physical molecules first. And my favorite thing about synthetic biology these days is that it actually works like Lego. So you have very simple bits, very simple blocks, and you can construct amazing things using those blocks. Imagine you have this protein and you want to introduce 3 different mutations into it. And then you want to have a library of proteins that carry a full combinatorial set of those mutations. All you need is the original protein and then the subset of mutations. Then you mix them up. I mean, you add a bunch of other reagents, but let's stick to this. And then you end up with a transparent liquid, with a tube with transparent liquid that contains a library of proteins that have the full combinatorial set of all these mutations. Once you have the tube with all of the physical molecules, you have to test those molecules. You have to rank them. And I said a couple of words before already about how we treat our tests. So we try and abstract away, and we try to implement those tests in a form of filters. The reason why I say filter is because we're not only interested in the output library, which is normally the case for drug discovery platforms, but we're also interested in the input library, because we want to compare the abundances and normalize everything by all of the biases in the system, and also want to get the negative data, right? Because negative data is extremely important for machine learning. Up until now, though, everything that a human scientist could see is this. They had a tube with transparent liquid before the filter, and they're still with a tube with transparent liquid after the filter. So the next step is to make the data computer readable. And we do this using the so-called next-generation sequencing machines. The reason why they're called sequencing machines is because they're capable of transforming the physical molecules into sequences, so strings. And the reason why they're called next-generation sequencers is because they're capable of reading in millions of physical molecules into the computational world. So now we have millions of millions of strings, and we want to process the data. The way we process the data, we have automated all of the steps because it's more or less similar across different projects. So we have our sequencers here, and then automatically, as soon as the run is done, everything is being uploaded to Google Cloud Platforms, and then we have automated all of the pre-processing, and then our data scientists are notified as soon as this process is done, together with, they also get a report with lots of different insights into the data. And in the end, they basically end up with this sort of data set, lots of different variants of a protein, and then the abundance score, the relative abundance score, so the fitnesses of these molecules. So the only thing we need to do now is to model the data. So we want to produce a model that takes in a sequence and spits out a number, right? Just to remind you, what do we need machine learning here for? If we have tested everything, like what do you need machine learning? Well, actually, we haven't tested everything. And experiments are so expensive and time-consuming that you want to reduce the amount of time you spend doing that as much as possible. So what we have so far is like different dispersed dots that we have sampled from the fitness landscape. And what we want to do, we want to use machine learning to reconstruct the mathematical function that links the sequence space to the fitness space. What would be the easiest case scenario? The easiest case scenario would be if every single position had a linear impact on the fitness. So we would just basically, one hot encoder of data would have a very simple two-dimensional matrix where each column corresponds to a mutation, and each row corresponds to a protein variant. And then we use a simple linear regression, and it could assign weights to our mutations. We know it's not like that, though, right? We know that the protein, the linear shape of the protein, folds into a three-dimensional structure, which means that the positions interact with each other. So the relationship between fitness and function is, fitness and sequence is not linear. So we need to add nonlinearity terms to the equation. And there are basically two ways of doing this here. Simplification. You could add interaction terms, so pairwise, triplewise, quadruplewise interactions. This will, of course, explode the number of features that we have in our data. Or we could add a nonlinear function that is wrapped around the linear combination of mutations. And evolutionary biologists have been thinking about this for quite a while, and they called the linear combination of mutations here a fitness potential. The reason is you have a linear variable, basically, that maps your sequence space to the fitness space and determines what is the fitness potential of the sequence that you have. And if you want to link sequence to function, what you want to do now, you want to simultaneously optimize the weights of the mutations and the nonlinear function. And it's a beautiful way of, I think, implementing this simplest feedforward neural network, because the first layer will give you a linear combination of mutations, and the last layer, so the rest of the layers will basically layer up the nonlinearity to your equation. Of course, it doesn't have to be just one linear combination of mutations. You can have many more than one neurons in the first layer. But actually, even if you have two, I'll just give you an example. Yeah. So this is a result of an experiment that we did with Kondrashov lab. And we had a protein and we tested lots of different variants of that protein. And then we had the same sort of data. We have lots of different variants and then the fitness scores. And then we used a very simple feedforward neural network with only two neurons in the first layer. And there's the results of those neurons are basically extracted here. So the two axes at the back, at the bottom, are telling you the linear combination of mutations. And then you have fitness here. In red, you have the measured fitness, and in black, you have the predicted fitness. So each dot corresponds to a protein variant. And you can see already that the accuracy of prediction is quite nice. And this is telling me two things. Thing number one is that you don't need a very complex model to describe the fitness to sequence relationship if you have a good enough sample of the empirical fitness landscape. And thing number two is that the interaction terms that I was talking about before are not going to be a concise representation because all of the positions interact with each other. You can see it here. In order to determine exactly what is the fitness of your molecule, you have to know all of the positions of the fitness of the sequence. Now, finally, you have the several models that you have optimized for different properties, and you want to optimize the molecule, the original molecule that you started with. So we use, as I said before, we use multi-objective optimization in the form of in silico evolution. So we basically co-optimize different properties simultaneously. And then we, so it goes literally like in silico evolution, it goes every single step. You select the fittest variants from the Pareto front, and then you make mutants from those variants every single iteration as well. And in the end, we end up with a subset of variants that we can incorporate in our next generation library designs or test on a lower throughput scale, which I'm going to talk about next. So the next step is to test the prediction. Biology is beautiful and high throughput methods are amazing, but the drawback of high throughput methods is that they don't tell you exactly the different aspects of a molecule. So you have to go on a lower level, on a lower throughput scale, and you have to characterize the molecules that you have developed in order to prove that the models that you have built are actually correct, that they're an accurate reflection of the real life. So for that, we use E. coli, bacteria that lives in our guts, and we have a whole protein team that is devoted to that. So as soon as we have a subset of candidates, around like 100 of different candidates, we go to the protein team and we say, we need this, 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 and this feature tested. Then they make these bacteria to produce as much protein as they need. Basically, they treat them as protein production machines. And then we have different assays that are developed specifically to characterize the different features of a protein on a lower throughput scale. And data scientists are treating this protein team as a very expensive hypothesis testing technique. So once we've done that all, we want to eliminate humans from the platform as much as possible, and we want to automate. We have a whole automation team among us, which is 30 people, so it's like a fairly small team, but it's headed by an engineer from Volvo. And as soon as we feel like our platform, different algorithms that we have established in the lab are robust enough, they come in and they automate it so that our scientists have the time to be creative and develop new capabilities to the platform. And meanwhile, the platform can be working 24/7, which humans don't normally do. And also, we have full transparency over the things that have happened in the lab. So if you have missed the majority of this talk, I'm going to summarize it for you. At LabGenius, we are developing a platform to design new proteins. And we use for that synthetic biology, machine learning, and robotics. We use synthetic biology to take the metaphorical GPS trackers and to drop them onto the fitness landscape and to record the coordinates of the fall. Then we use machine learning to reconstruct the shape of the fitness landscape and in silico evolution to crawl up those peaks to sample from those peaks the new molecules. And then finally, we use robotics to automate the platform and make it learn every day from the data that we produce. And of course, there is a myriad of fantastic and exceptionally smart humans behind this platform. These people come from very different backgrounds, which is a very challenging thing in its own to establish the communication. But it's a different talk. They come from different nationalities as well, I believe about 14 nationalities, and I'm a massive fan of every single one of those. And finally, we're hiring. We're growing quite rapidly, and you should pop up our website and see the jobs. Thank you very much.